All right, today we're diving into the powerful science of genome-wide association studies. You'll often hear it called GWAS. It's a technique that has absolutely transformed how we understand the genetic roots of, well, pretty much all human traits and diseases.
So let's kick things off with a question that people have been asking for forever, really. Why do complex diseases like schizophrenia or heart disease seem to run in families? What is the genetic link there?
To figure this out, scientists have basically had to become genetic detectives. They're on this massive hunt, searching for tiny clues hidden away in the 3 billion letters of our DNA, trying to figure out what makes some of us more likely to get certain conditions than others. Okay, so if they're detectives, what exactly are these clues? And how on earth do you even start looking for them across the absolutely vast landscape of the human genome?
Well, the main clues they're looking for are called single nucleotide polymorphisms, or much easier, SNPs. The best way to think of them is just as single-letter typos in our genetic code. You know, where most people have a C, maybe you have a T. These tiny little variations are what make us all unique, and they are exactly what our genetic detectives are hunting for.
Now here's where it gets really tricky. For most of the complex traits we care about, there isn't one single smoking gun gene. Instead, it's more like this quote says, there are thousands of these tiny SNP clues involved, and each one on its own has a tiny, almost unnoticeable effect. So that creates a huge puzzle, right? How in the world do you find thousands of minuscule clues scattered all across the entire genome? Well, it turns out our detectives have a seriously powerful tool to help crack the case. They use a technique called a genome-wide association study, or GWAS. And you can literally think of it as a massive, unbiased search party. It scans the entire genome, comparing the DNA of thousands upon thousands of people to find those all-important SNPs.
And the core idea is actually pretty straightforward. Scientists get two big groups of people. First, you have the cases. These are the people who have the disease or trait. And then you have the controls, the people who don't. Then they just compare the frequency of every single SNP typo between those two groups. If a certain typo shows up way more often in the group with the disease, boom, that gets flagged as a potential clue.
But, just like any real crime scene, the raw genetic evidence, it's messy. It is full of errors and random noise that can easily send detectives down the wrong path if they're not super careful.
And I'm not just being dramatic here. Failing to properly clean the data, to really scrub it, has led to huge, high-profile scientific papers being pulled back, retracted from major journals. That shows you just how absolutely critical this next step is.
And this is where quality control, or QC, comes in. Think of it as the rigorous, painstaking forensic work. The detectives have this whole checklist. They're looking for everything. Technical problems like missing data or errors from the lab machines. Even sample mix-ups, which, believe it or not, they can spot by checking if someone's recorded sex matches their genetic sex chromosomes. They also have to weed out things like hidden relatives in the data, because that can totally skew the results. It's all about making sure they're chasing real genetic leads and not just some technical glitch.
Now, one of the biggest, sneakiest red herrings that can fool even the sharpest detectives is something called population stratification. This happens because those little SNP typos can just naturally be more or less common in different ethnic groups. And if you're not careful, you might find a false clue that has absolutely nothing to do with the disease you're studying. And this plot right here, this is how scientists actually see that problem. It's so cool. Each little dot is a person, and you can see them naturally grouping together based on their genetic ancestry. By mapping this out, researchers can adjust for these differences and avoid false alarms. There's a famous case where a study incorrectly claimed to find a chopstick gene. Turns out, it was just an artifact of this exact problem. A huge lesson for the whole field. Okay, so after all that incredibly hard work of cleaning the evidence, we finally have a list of real, trustworthy clues. But that just leads us to the next big question in our detective story. So remember how we said that each individual SNP has just a minuscule effect? So what do you do with that? How can you possibly combine thousands of these tiny, almost insignificant clues to build a meaningful case about a person's actual risk? The solution is actually really elegant. It's called the polygenic risk score, or PRS. The idea here is instead of focusing on any one SNP, you aggregate the effects of thousands, sometimes even millions of them. You put them all together into one single, powerful score that represents a person's overall genetic liability. And here's basically how it works. For any one person, the score takes the effect of every relevant SNP they carry. It multiplies it by how important that SNP is, its weight, and then it just adds them all up. What you get at the end is one single number that captures a person's total genetic risk for a specific trait. So, with all that said, let's just step back for a second and see how this whole buildup of evidence, from that first giant search party to the final score, has truly revolutionized our understanding of genetics.
Let's put it all together. By combining a massive search party, that's the GWAS, with meticulous forensic work, that's the quality control, and then adding up all the tiny clues into a polygenic risk score, we can finally get a much, much clearer picture of someone's genetic risk for all sorts of complex diseases.
And the results? They've been staggering. For instance, using these exact methods, scientists have now identified over 100 different genetic regions associated with schizophrenia. This has just blown the doors wide open on understanding the biology of the disorder, and it's something that would have been completely impossible just a few decades ago.
And that leaves us with one final and pretty provocative thought. As these polygenic risk scores get more and more accurate, what's it going to mean for us when our genetic risk for hundreds of diseases can be calculated from just a single drop of blood? What does that future even look like?